Hi, Assalamualaikum and good day to Doctor. So we are from Group 6. We will be presenting our mini project, Microbe, R, Anissa, Kuganeswaran, Lizishwan, Fazlina and also Vishalini. So what is food contamination? Food contamination refers to the presence of bacteria or fungi on the food, especially fungi or harmful chemical in the food. This contamination can cause illness to the person who eat the food. The contamination can occur when the food is exposed to the environment in which the bacteria can easily grow. For our topic, cake contamination is referred to the mold but also consists of some species of yeast and bacteria which infect the cake. This is because of the increase in sugar content which lead to the formation of cake spoilage.
Okay, so for the problem statement, as a quality control executive in the food industry, the detection and the quantification of bacterial contamination in cakes are critical for ensuring food safety. Traditional methods for measuring bacterial growth may have limitations in terms of speed, sensitivity, or specificity. Therefore, there is a need to explore advanced scientific methods to enhance the accuracy and efficiency of detecting bacterial contamination in cakes with a focus on maintaining product quality and also meeting regulatory standards. Isolation of microbial. There are two methods for isolation. One is spread plate, another one is pour plate. So for the spread plate, first we need to obtain the sample. Then we will do a serial dilution. Why we need to do a serial dilution? Because the concentration of bacteria in the sample is too high. So the serial dilution will help to reduce the initial concentration and making it feasible to count colonies on the agar plate. Then after that we will pipette the bacteria sample onto the surface of the agar plate. Then we will spread the sample evenly over the surface by using a sterile spreader. Then we will incubate it under suitable condition. Lastly, we can observe the colonies grown on the surface. So for the pour plate, first we need to obtain the sample, then we will do a serial dilution. After serial dilution, we will pipette the bacterial sample onto the petri dish. Then we will pour the liquid agar into the petri dish. We will swirl the petri dish to mix them. Then after that, we will incubate it under suitable condition. Lastly, you can observe colonies grown on the agar surface and also subsurfaces.
Thank you, Visha, for the presentation. I am Li Zixuan. Now I will explain the analysis method of microfoam cake using biosensor strategy. Biosensor is the modern technological analytical device that functions to analyze a sample in the presence of a specific target analyte. In our case, sample is cake and analyte is microbe like bacteria. As shown in the diagram, biosensor has two major components which are bioreceptor and transducer. Bioreceptor is the biological component such as enzyme or antibody that play a role of interaction between the analyte resulting biorecognization. This biorecognization will be converted into a measurable signal by transducer. So why you want to choose biosensor to detect microbe growth from the cake? Here are some reasons. Firstly, biosensor is highly sensitivity, fast processing, and give real-time result. By using the conventional method like molecular analysis using media such as spread count is time-consuming and low sensitivity due to contamination can happen during analysis. Sample preparation is needed in conventional method and this will not give the real-time result at all. Next, biosensor is highly specific to detect or determine the microbe from the cake. Besides that, the sample used in the biosensor is small amount only and can directly use due to no sample preparation is needed. Moreover, biosensor can detect all types of microbe that cannot be carried out using culture media analysis, since not all bacteria can go on culture medium. Lastly, biosensor technique is simple and easy to be carried out, hence it's faster and more effective, while resulting in cost-effective method. There are three types of biosensor that play role in bacterial detection, which are optical biosensor, mechanical biosensor, and electrochemical biosensor. First, optical biosensor will exploit the analyte binding induced change in the optical properties of the sensor surface, which are then transduced to a detector. The optical biological sensor contains a biodegradable polymer by analytical enzymes secreted by microorganisms during the deuterization of the natural product. Surface plasmon resonance (SPR), as shown in the diagram, is the most common biosensor used to detect the microbe growth from the cake. SPR system comprises of a source of plain polarized light which then pass through a glass prism, the bottom of which contains the bioreceptor functional light transducer surface, which is typically a thin film of gold. Analyte binding to the transducer surface changes the prism refractive index, which in turn alter the angle of the light exiting the prism. Example of the bioreceptor in this biosensor can be used are antibody, bacteriophage, and lectin. The second type of the biosensor is mechanical biosensor. It is able to measure a mass-sensitive sensor surface diffraction because the target analyte will be bonded on the functionalized surface. It is useful in microbe detection since it can provide high sensitivity and quick processing time without the need for sample processing or extra reagent. One example is quartz crystal microbalance QCM biosensor, as shown in the diagram. QCM will detect the resonance frequency change that result from increased mass on the sensor surface due to analyte binding. The last type of biosensor is electrochemical biosensor. Usually in electrochemical biosensor, biological reaction either generate a change in signal for measured curve which might be measured by impedimometric technique. Using impedimometric biosensor technique function by analyte bioreceptor interaction causing a change in capacitance and electron transfer resistance across a working electrode surface. As analyte binding increase with higher analyte concentration, the independence across the electrode surface change and it is detected as a transducer. So that's all for my part. I will pass to Anissa for further presentation. Thank you.
Hi, I am Anissa and I will explain the next method. The method that will be used to measure bacterial growth in bacterial contaminated cake is dynamic method. This method is started by preparing 9 different cakes from a bakery, which are 3 sponge cakes, 3 mousse cakes, 1 cheesecake and 1 tiramisu, and also 1 brownie. The cakes were stored at 4 Celsius until it was analyzed. All cakes will be mixed before analyzing. Proximate compositions such as moisture, fat, protein, and carbohydrate content and water activity were measured to determine the effect of the growth of Salmonella species and S. aureus in a cake. The water activity of the cakes was determined using a water activity meter. 6 strains of Salmonella species and 5 strains of S. aureus were prepared and stored at -80 Celsius and were cultured in 10 ml of tryptic soy broth at 35 Celsius for 24 hours. A mixture of 6 Salmonella strains was used as an inoculum, and also a mixture of 5 S. aureus strains were prepared. The cakes were cut into cubes and placed in a plastic container. 0.1 ml aliquots of the inoculum were evenly seeded in the cake samples. The inoculated samples were stored at 15 Celsius for 7 days for S. aureus or 9 days for Salmonella species. To enumerate Salmonella and S. aureus cells, each cake sample was transferred to a sterile sample bag and treated with 50 ml of saline solution, followed by pummeling with a pummeler for 1 minute. The samples were then serial diluted with saline solution, and 0.1 ml of the diluted was surface plated on xylose lysine deoxycholate agar and Baker agar for Salmonella species and S. aureus respectively. The plates were then incubated at 35 Celsius for 24 hours, and the colonies were manually enumerated. Cocktails of Salmonella species and S. aureus were inoculated in a selected cake sample as a model. The inoculated samples were stored at 4, 10, 15, 20, 25, and 30 Celsius for Salmonella species, and 4, 10, 15, 25, and 30 Celsius for S. aureus. The cell counts at each temperature were fitted into the variant model. There will be 2 formulas used for predictive models for isothermal temperature. The first one is the binary model. It is a model whereas the cell counts of each temperature will be fitted into the binary model using DMV3.5 to determine the maximum specific growth rate and the lag phase duration. As for the second formula, it is the RMSE formula. It is to validate the predictive model developed, where additional experiments were performed at 23 Celsius and observed data were compared with the predicted data obtained from the predictive model developed in this study. This is an example of the data that can be collected from the binary model and RMSE. To describe growth patterns of Salmonella species and S. aureus in the cake sample at different temperatures, a time temperature profile of 10 to 25 Celsius was maintained using a programmable refrigerated incubator, and Salmonella species and S. aureus cell counts in cake were also measured under changing temperature. The temperature profile was recorded using a data logger and used to predict the bacterial growth patterns with the equation reported by Bailey. For dynamic model development, a secondary model for Umax will be developed for isothermal temperature, was used for calculating Umax under changing temperature in the recorded data logger, followed by applying the binary equation to estimate Salmonella species and S. aureus counts in real-time prediction. H0, the value meaning potential growth, was obtained from the average of multiplying Umax and LPD of the perimodel at each temperature. To evaluate the performance of the developed dynamic model, RMSE was calculated. As you can see, this is the dynamic model for the Salmonella species and the S. aureus in the cake during changing temperatures.
Hi, I'm Google. Now I will explain how real-time PCR help us to quantify bacterial contamination in cakes. First of all, real-time PCR, also called as quantitative PCR, is a PCR-based technique that couples amplification of the target DNA sequence with quantification of the concentration of the DNA species in the reaction. Now, let's see the steps in real-time PCR. The first step is DNA extraction. DNA will be extracted from the cake sample. This DNA represents the genetic material of the bacteria in the cake. The second step is primer design. Specific primers are designed to target specific regions in the bacterial DNA. These primers will only amplify DNA from the target bacteria, providing specificity to the analysis. Third step is real-time PCR setup. A real-time PCR reaction is set up with the extracted DNA, primers, DNA polymerase, nucleotide mix, and other necessary components. Fourth step is PCR amplification. The PCR machine amplifies the bacterial DNA present in the cake sample. As the reaction progresses, the fluorescence increases and the machine records the fluorescence at each cycle. Fifth step is data analysis. The real-time PCR machine generates a graph showing the increase in fluorescence over cycles. The cycle threshold CT values is determined, which represent the cycle number at which the fluorescent signal crosses a predefined threshold. Lower CT values indicate higher amount of initial DNA in the sample. The sixth step is quantification. The CT values obtained from the cake sample are compared with the standard curve generated using positive control with non-bacterial DNA concentration. This allows the quantification of the bacterial DNA in the cake sample. The seventh step is calculation of bacterial load. The amount of bacterial DNA is directly related to the microbial load in the cake sample. By comparing the CT values of the cake sample with the standard curve, you can calculate the bacterial load in each sample.
So in conclusion, the isolation process for microbial using two methods, which are spread plate method and also pour plate method. Furthermore, there are three types of solution where we use to measure bacterial growth in contaminated cake, which are biosensor analysis, dynamic method, and also real-time PCR. Advanced technology help in producing or reducing the contamination in the food with the various methods and procedure. This also help in the QC production to produce a good quality and of the product. So that's all from our group. Thank you.